no further ado on that one. So this is our new friend Alan. He is an expert in this specific topic. So he is the founder and executive director of the Institute of Integrated Regenerative Design. They train professional design practitioners to create systems that are focused on the regenerative and sustainable aspects. After starting with a degree in electrical engineering, he moved into systems engineering and large-scale systems designs. He now has over 35 years of experience in engineering and 24 years in sustainable and regenerative design. With deep hands-on experience designing large-scale sustainable systems, Alan is a lead app BD+C well-accredited professional. GRP which holds the Living Futures accreditation for working with the Living Building Challenge and Living Community Challenge projects. And he is also, as you know, the creator and one of the chief designers of the integrative design framework for the company, and they focus on, and the lead author of the biocompatible design standards. In addition to teaching permaculture and integrated regenerative design, he also provides consultations and workshops on earthworks, watershed management, bioremediation, composting, forest gardening, holistic management of pasture lands for the key line designs, aquaculture, and aquaponics. He also focuses on net zero buildings, off-grid energy systems, and natural building systems. Having worked with bioremediation for the past decade, Alan was asked to join the team for responding to the ecological crisis caused by the Lahaina fires in Maui in August of 2023. And over the past year, he has been helping figure out how to scale the bioremediation practices and address the large-scale disaster situations. So when we say that we are, very thankful and blessed to be here, we are sincerely, with our most warm, heartwarming, applauding for him coming here. So if you don't mind, I'm going to go ahead and transition him. Thank you so much for coming. And Alan, you take it away.
Well, thank you for being here. Congratulations to Aaron for getting through the letter soup of my credentials. Normally, I'm teaching design professionals, and so we have to wow them with all the letters and that kind of stuff so we can listen. It's just the way the game is played, right? But in reality, I think it's like, how many people here grow a garden or farm? Okay. I go in and I teach engineers, and I tell them, if you can't grow a garden, you can't engineer it. And they go, what are you talking about? By the time we get done, they know what I'm talking about. You can't grow a garden and you can't farm, you don't know how to engineer, right? We shouldn't let anybody be an architect for a building that can't grow a garden. That's my opinion after about 35 years of doing this, right? I was in, let me just tell you what we're going to do real quick, and then, because I know there's going to be some questions, so I'm going to jump right into it. Yes, ma'am.
Can you repeat the website that these slides are going to be on?
What we're going to do is this, that sheet that you have in front of you, all the concepts that I'm going to cover, the key concepts, are right on there, and we're going to go right down it, okay? I'm going to do it. We'll look and see if I can get these slides up, and we'll make certain we post it at the end where it'll all be, because it's setting up that website. So we'll get it for you, yes. So
we'll make an announcement at the end now.
Yeah, we'll make an announcement again at the end.
That all the content is recorded here today. So not only the video and audio, but also every PDF that he is doing with the slides will be available online, completely free. And we ask that everyone shares it.
So I will just note that I know somebody's going to ask this at some point. Now, Institute is a nonprofit. We do not accept any government funds, so I can say what I need to say. Okay. Just leave it at that. Okay. What I'm going to do tonight is we're going to do sort of Bioremediation 101. And I'm going to ask her in a moment. I want to know who all is in the room. So we're going to kind of take a poll of the room. But I know some people are going to show up. They're like, what is bioremediation? So we're going to start at the very beginning. We're going to get you up to speed on it, get you all the basic concepts and everything. And then after that, we're going to do a Q&A. So what I'd like for you to do is, during the presentation, during the 101 part, please keep any questions you have to exactly what we're talking about. We'll have a general Q&A at the end, okay? So if you need clarification on something I'm saying at any point, just throw up your hand. I'll try to catch it and we'll clarify what we're talking about, and then we'll have a general Q&A at the end. Then what we're going to do right after that is V is going to come and facilitate the process. Some of the folks that are doing bioremediation and other, ecological regenerative processes on the ground, we'd like for you to meet all of them. So we're going to go through that and have a little bit of time for everybody to kind of share that. And then at the very end, we're going to get into an opportunity to organize and connect. So if you're interested in how to plug in, if you're interested in organizing something, we're going to start trying to figure out how to do that because, I was joking with Billy when we first started talking about this. I said, I'm not into NATO meetings. He said, what's a NATO meeting? I said, a friend of mine, another engineer, came up with that. He said, NATO stands for no action, talk only. Okay. And see, it's like, I think Billy's like me. If we get in a NATO meeting, it's like hit the eject button and get out of there, right? Okay. So it's like, we got folks here that are going to get biology on the ground next week. Okay. And the reason I was coming over was like, let's get people on the ground set up to where they can actually start to do something and give you what you need in order to do that and answer your questions and get you going. Okay. So that's what we're up to. And with that, I'm just going to kind of jump in. Okay. And I want to jump in here. This is Lahaina in August of last year. Well, actually, there's about 8 square miles of this. Okay. I happen to be on the Big Island right after this with one of my other instructors, Chase. We were working with a number of the indigenous farmers on the Big Island of Hawaii, talking about their hydrological issues they were having because of the development. The way that the development's being done on the island, they're starting to have problems with water that they haven't had in the many, many, many generations that their families have been on that land, right? And so we were talking about that, and I got a call from the folks that are doing the Maui Bioremediation group, and they said, hey, can you help? It's a great group. We have mycologists in that group, bioremediationists, and so on and so forth. And they're like, we're going to need some help scaling this thing up, right? Because it's big. And so I jumped in and have been helping them, sort of go forward and figure out how to do this. And there's a lot of stories with this I don't have time for. We could go through hours of stories on this. But what I will tell you is one of the things we ended up having to do very, very quickly was we realized they were putting filter sock out. And we said, what really you need, you basically, these silt socks. You may have seen those for like job sites and so forth to try to keep the silt out. Yeah, they were putting those out because what you had was 8 square miles of toxic ash right uphill from one of the world's most beautiful and biodiverse marine reserves. Like what you don't want is all that ash going down into the ocean, right? And so what we started doing was looking at how to take, and right after the silt sock, we put another sock that was a biofilter sock. That had a combination of mycelial inoculated wood chips and biochar and a few other little things, and put it right after that. And what you kind of see is that whereas the filter sock would kind of, the silt sock would kind of stop a lot of the particulates, that what we were doing right after that was actually capturing a lot of the dissolved chemical contaminants kind of before it got down. So, with that, both the combination of the mycelium, the bacteria, and the biochar. Okay, so that was kind of we jumped in and started doing that. And then one of the things that is true about working in an island ecosystem like Maui is they're very sensitive to having you bring in things that are not indigenous, right? Because their ecosystems are you bring in things that, boy, it's hard to know exactly what it's going to do. So we decided that early on we were going to use all what's called indigenous microorganisms. So everything we have used in remediation has actually been indigenous to the island. What you're looking at here, for any of you who are familiar with this, is a technique called IMO gathering. This is This is white rice that's taken out into intact ecosystems and allowed to actually gather mycelium, mycospores that are coming from intact ecosystems. And we actually culture those out and figure out which ones of those can be used to help break down nasty stuff. Right? So that's the sort of stuff we're looking at. And then out of that, we started spinning up a nonprofit called EarthRM. So what we need to do is we're generating intellectual property here. And if we don't take it and protect it, somebody else is going to try to grab a hold of it, patent it, and commercialize it. Like what we need to do is spin up a nonprofit that will hold that intellectual property necessary to do what's called defensive patents, which is patented so that we can then license it freely to all the communities that need to use it. Instead of having it, then if anybody wants to use it commercially, then you could, they can license it for commercial application. We don't want it locked up when people like here in this area can't use it, right? All right, so that's kind of what we ended up doing, and we're creating educationals and so forth to go along with that. So, boy, like I said, there's a lot of stories we can tell there. What I'd like to do is ask this real quick. Let's see how familiar everybody is with the idea of bioremediation. We'll ask you to identify as one of four groups for me. One is barely heard of bioremediation, and I really don't know much. Number two is I'm familiar with the concepts, but have never really done it. Number three is I've done it to some degree. And number four is I'm really experienced with bioremediation, leading projects, and so on and so forth. So let's just kind of do that. First of all, number one, I'm just barely, know the basics, basic concepts and so forth. Okay, very good. Number two, I'm familiar with the concepts. Yeah, that's a good, healthy dose of people. Good, so that's maybe the majority of people. Number three is I've actually done some bioremediation on the ground. That's impressive. Most of the areas I go to, the answer is maybe one or two hands, and I figured it here in the Asheville area we would probably have a lot more than that. And then number four, people who are like really on it and have done. We tried to get some of them down here at the front just so they can. We're going to have all of you guys introduce yourself at the end and talk about your projects when we get done with the 101. Okay. So we're really glad everybody is here because it'll take all of us. Everybody from the ones who barely have ever heard of bioremediation all the way through the experts, all of us together to attack something of this scale and start to get it done. So what I'm going to do is I am going to do a bioremediation 101, which means I'm not going to get into like this specific strain of, fungal, spores, what we want to use for this, or this particular plant for that. What I'm going to do is get you the big picture concepts and then talk about big picture how to attack the problems that we're seeing. Then when we get into actually on the ground, we can start getting into those details, right? But here's the interesting thing. We talk a lot about which species of fungi and so forth. You realize that we got some of Billy's Biocomplete compost here. 1 teaspoon of that probably has 30,000 species of fungi in it, right? It probably has I want to say 60 to 70,000 species of bacteria, but I would actually argue that the idea of a species of bacteria is kind of a nebulous concept. Any microbiologists in the room? Yeah, Lynn Margulis argued the concept of a species because the whole thing is they actually do this thing called horizontal gene transfer. They actually give each, so if one of them figures out how to solve a problem, they share with everybody else. And next thing you know, they all know how to do it. Okay, so what that means is this idea that we have to find the right. It's like let nature. They're the experts. We'll sort it out. We get 40,000 fungi species in there, and guess what? They're going to start figuring it out right quick. Okay, so the good thing is we can just get really good biodiverse stuff on the ground, and it'll go to town, right? It will, and so you don't have to know how to sequence, DNA and all that kind of stuff to do this. It's very practical. So let's go ahead and get into the basic terms and concepts, right? Okay, I'm just going to call bioremediation partnering with living organisms to clean up toxins and pollutants in an environment. Just that simple. It turns out that life has figured out this stuff, and has a lot of different ways of doing it. But there's a couple of more specific terms that you'll hear used in the literature. The first we would call microbial remediation. And this is using microbes, most often bacteria, to perform bioremediation, okay? For those of you who are a little more, there are a few cases where archaea come into it, but in most cases we're talking about bacteria. We talk about microbial remediation. And it turns out that a lot of the ones that we're working with are in the Cocci family and certain of the Bacilli family. A lot of those are the ones we end up working with a lot. And then some of them are in the not so happy groups and that we don't want to see a lot of, like some of the Vibrios and things like that. So we look at them under the microscope and we can usually see general families that when we do microscopy, okay? The third one we want to talk about is phytoremediation, and of course phyto means plants. So you put phyto in front of it, and it means the plant does it, right? So obviously phytoremediation is plants doing bioremediation, and the whole thing, of course, is that the scientists love their words, so here they are. You put phyto in front of all the words, here they go. Phytovolatilization, which is we basically get the plant to uptake these volatile compounds and transpire them out their leaves up into the atmosphere, right? You get photodecomposition. So photodecomposition is something different. That's when sunlight breaks it down. Phytodecomposition is where the plant facilitates the chemical breakdown of complex organic compounds into simpler harmless things. There's phytoextraction, where the plant takes it up into its body, right? And then if it's something like a heavy metal, we can then harvest that and get it off the site. Phytostabilization, sometimes called phytoimmobilization, is where the plant helps to lock it up and so that it is chemically inert. And then phytostimulation is where you get these beautiful, sun-gathering leaves up in the air. They start to create sugars and such, and they go down and it become exudates out of the roots. They stimulate microbial response. We call that phytostimulation. Okay, so those are all different ways that we can start to work with plants in order to start to clean up things. Then mycoremediation, right? Myco is fungi, mushrooms, but, well, of course, mushrooms are a fruiting body, right? So, but just, so micromediation is using fungi to perform bioremediation. So those are some of our just base terms and so forth. And I want to hit this one, microbiome. So what is a microbiome? It is the community of microbes that live in a specific environment, such as in the human gut. That's the human gut microbiome. We also have a mouth microbiome, a skin microbiome. We have a lactation microbiome. And of course, a tree. You realize there's actually a fancy word for the tree plus all of its microbiome and all of its symbiotes. It's called a holobionte. Because, how people say, we need to plant 1 million trees. I'm like, no, you need to plant 1 million tree holobionts. Because a tree without all of its symbiotic microorganisms is not a happy tree. Right? So we need to think about how to do that, right? So it's like, yeah, all right. And by the way, what are these? What are these guys? Those are spirochetes. Do we like that or not when we see them under the microscope?
No.
No. Those actually are typically indicative of what kind of conditions? Anaerobic conditions. Yeah, exactly. Okay. So there we go. I'm going to go fast. This group's a little bit ahead of the normal curve. Okay. Keep on going. Biological inoculant. Okay. Again, here's another one of the IMOs that we did in Maui. Okay, it's a source of beneficial microorganisms that can be used to establish a healthy population of microbes in the environment and helping to clean up contaminants and reestablish a healthy environmental microbiome. So since we're a little bit, this group's already familiar, I want to talk about this a little bit. I'll jump a little further in. Okay. What I would say is that the researchers have found that there are nested microbiomes, and that every microbiome that's nested inside of a larger microbiome is heavily influenced by its overarching microbiome. So the environmental microbiome of an area would be the overarching microbiome. And then there are niche ecosystem microbiomes that go all the way down to microbiomes inside of little areas inside. They're all nested inside of each other, okay? The whole thing is it's very difficult to maintain healthy gut microbiome if the environmental microbiome right around you is actually profoundly unhealthy. So one of the things that we know is that human health, when we obviously talk about all the physical, the chemical contaminants that just got dumped all over everything. But there's another whole thing for us to think about if we want to be healthy, which is what just happened to the environmental microbiome, right? What did that just do? And what's that going to do for everybody who lives in that? And by the way, of course, humans are not the only ones that have a gut microbiome. How about ruminant animals? All these kinds of things. So there is a huge environmental cascading effect. And so what we can do is we can find places that still have healthy environmental microbiomes. We can harvest from that indigenous, that's what should be here, right? Not bringing it in from somewhere else, not buying it from anybody and bringing it in from California or whatever. We find the good microbiomes that are present here and we help reestablish them. That's what we should be up to, okay? So we use those as biological inoculants. And of course, one of the first things that we start thinking about is different forms of compost. Now, don't have time to get into the full depth of it, but we would say that there are many different kinds of compost, of course. Most people are more familiar with thermophilic compost, which is your hot pile compost. And when that's done right, it can be very biodiverse. It's a little more difficult to get it to be very fungally dominant. It takes a little more skill. But it can be done. And then there are other forms like Johnson's bioreactor and so on and so forth. We won't get into all of it right now. We'll just say that all of these, when it is done right, should be inoculated with local biological inoculant. And it should be very biodiverse. And that then becomes an excellent source for us to look to in order to be able to do a lot of the bioremediation that we're going to do that's microbial and fungal. Okay? So I get into places and I have people who are experts at doing compost and so forth, and they go, well, I don't know how much help I can be. I'm just a farmer. And I'm like, if you can do compost right, you're of more help to me than a dozen PhDs. Okay? Seriously, right? Because if you can get good biodiverse life, microbial and fungal life going, then you've got the raw stuff we need in order to do the microbial and the mycoremediation, and to get the phytoremediation jump started, because that's the stuff that makes up the soil food web that is symbiotic with all the plants we want to use, right? So what we're looking for and what we need is we need the seeds of the phytoplants that we are going to work with, and we need the bacteria, and we need the fungi. And it turns out that people who know how to do really good compost, that's what we need, okay? So, good compost is alive. I need to be able to look at it under a microscope and see what all is there. We'll do what's called qualitative microscopy, and that's looking at the size, shape, and color of all the microorganisms in there. We'll figure out what our general balance is of good guys and bad guys. And the quantitative side is counting the number of those per field and getting down to being able to quantify how alive is that. It ought to be aerobic. If it goes anaerobic, please repeat after me. If it's anaerobic, it is not compost. It's actually a fermentation. Now I know somebody out there is going, oh, wait a minute, Alan, there is anaerobic compost. And what I would say is, okay, that's a colloquial statement. There are different ways of doing what we would call an anaerobic fermentation. Okay, that's technically what it is. And that can be helpful, by the way. But typically when we do the anaerobic, what do we do right after that? We do an aerobic stage to get us back to the good biology. Okay, there are times when we use anaerobic fermentation and anaerobic decomposition to break down things. But then after that, before we apply it, we're going to take it back to an aerobic stage. So we get the good biology back, right? A friend of mine, she was called out. She's in the Alaska Department of Natural Resources, and they called her out to take care of a whale that had beached. And she's like, what do I do with this? It was a pretty darn good sized whale too, 45 feet or something. So she actually buried it and anaerobically composted it. And then took it out after 2 years and went through an aerobic stage. Had some of the richest stuff you ever wanted to see. So yeah, you can do that. You can anaerobically decompose a whale into something that you can then do an aerobic cycle. And then you got good stuff. You can grow great crops with it, okay? But in general, for what we're talking about, if it's anaerobic, it's not compost, it's not got the stuff in it we want, okay? It needs to be nutrient rich and it needs to smell like rich forest soil. What happens is when you get into anaerobic decomposition, you get all kinds of wonderful byproducts like putric acid, which smells like vomit. That's why it's called putric acid, right? There's a bunch of other stuff. Well, hydrogen sulfide compounds come out of anaerobic. So the stuff we want is going to smell really great. And so Billy's brought along some. We're going to, that is here. You can come up afterwards, take a look at it. I mean, it's amazing stuff.
That's regular, the other stuff's vermicompost.
That's the vermi right there, right? We're going to get to vermi in a minute. So yeah, this is amazing stuff. And Michelle does the microscopy on it. This is what Dr. Elaine Ingham calls biocomplete. It has all the good guys in it you want to see, which is all the bacteria, all the fungi, all the good protozoans, all the good nematodes. Probably seen some really good microarthropods in there. Yeah, all the goodies, right? They're all in there, okay? That means that's gold in terms of like being the biology we need to get things going, okay? So yeah, on the right there, that's not compost. That's anaerobic. And so for what we're looking for, not good, okay? Yep, we do use a microscope. It's actually the most effective and efficient way of doing it. I'm kind of curious, how many of you have done soil microscopy? That's again, pretty good number of people, right? It's actually, I've taught high schoolers to do it pretty quick. Now, to get good at it takes a while. But like just to get the basic idea of it and so forth, I've done, afternoon workshops with high schoolers and they get it. It's not that difficult. And by the way, the microscope you need, you can get for $300, $400. It's not a multi-$1,000 piece of equipment, right? Now, yeah, you can spend more if you want to go higher end, but you can get the basics for $300, $400, okay? By the way, for those of you looking at it, we always use a 10x eyepiece. I happen to like a 4x, a 10x, a 20x, and a 40x objective, because we really don't need to go over 400x magnification. I'd cheat and have a 60x, because my eyes are not quite as good, but you don't really need anything more than a 40x objective. You're looking at 400, we do a lot of work at 400x, right? A lot of 400x magnification, okay? Vermicompost, right? Who does vermicompost? Wow, that's amazing, right? Vermicompost. So what's really interesting is if you look at the biology that's in vermicompost and compare it to the biology that's in a thermophilic compost, you're going to find that they're very complementary. There's stuff the gut of these vermicomposting worms are little bioreactors that have certain biology in it that's a little harder to find in thermophilic compost. And so when you mix thermophilic and verma, you get an even more biodiverse thing. So it really works. So, the vermi is mixing vermi, and then if we have a Johnson-Su, that's usually very fungally dominant. And what you see is after the Johnson-Su has gone through about 8 months, the fungal population and the biodiversity of fungi somehow manages to go up, which is really interesting. By the time you've had a Johnson-Su bioreactor for a year, the fungal biodiversity is amazing. So, that's another thing we can be looking at. Anybody who's got a Johnson-Su that's a year old? Yeah, imagine. We need to look at that. That's like that. You might have some magic going on there. Okay? Yeah. So what we always do, and Billy's got some great vermi here if you want to come look at it. And I always take all of it and sieve it. I always sieve it before I use it, especially for what we're going to talk about here, because As you're sitting here thinking about it, if you think about how much compost it takes to go on your garden, you're probably sitting there thinking, Alan, we got square miles of land we got to fix, right? How in the world are we going to do that? We're not going to put a half-inch compost on all that. Anybody want to build that compost pile? Right? So we actually have to have a way of taking that compost. We're going to have to create a bloody lot of compost to clean this up. However, we have other ways of multiplying this compost and actually getting it out there. That's what we're going to get into next. Okay? So, compost extract. I'm going to have Billy come up.
Tea time.
It's, well, not plug tea. Tea is next. This is an extract. Okay? So, I want to make a differentiation between an aerated compost tea and a compost extract. We're going to make a compost extract right here in the room. We could not make an aerated compost tea here because we wouldn't be here that long. How long does an aerated compost tea typically take? 24, in some cases 48 hours according to what you're doing, but usually it's a little over 24 hours, right? So what we're doing with, and this we can teach anybody to do, what we're looking to do here are things that we can take, and we're talking about how to let some of the experts be able to generate lots of compost and then in central locations and then get that out to where people can use it and we can train people in the communities to be able to do things with it once the biology is there. So go right ahead. So what he's going to do is he's going to take some of his thermophilic and some of his vermi. He's going to put it into the bag. What's your strain size here?
400 microns.
400 micron strain he's got here. He's got some non-chlorinated water, and all he's going to do is put it down in there, and he's going to just physically agitate. And by doing this, now, you can take, if you've got well water on a pressurized, you can spray through gently. You don't want to do it too strong, but if you don't have that, you can basically do what he's doing. What's happening is a lot of the humic and fulvic compounds are coming out into the water. Okay, at the same time, a pretty good amount of fungal hyphae and a lot of bacteria are getting into that water. And what we're doing is we're actually creating a water-based inoculant. So we've just taken, now the thing we can do with this is we can now take that and dilute that. Okay, so what we end up doing is, and we're going to talk about aerated compost tea here in a second, because aerated compost tea is harder to make, especially to do it right. However, that aerated compost tea is even better than the compost extract. Okay, I'll explain why in a moment. But if all you have is people being able to make a compost extract, then dilute it down and then spray it, you're in business. Yes?
Why can't you just put the compost in the water?
We don't, what we're going to do is we're going to put this into a spray pack. So we don't want a lot of big particles in there that will actually clog up the spray mechanism.
It won't just settle.
It kind of does and it kind of doesn't, because when you start to pump it and everything else, it's like, eh. So we want, yeah, so it's really better to do it this way so that we can, now, there are people who take this and then use a watering can. Okay, fine, right? That works too. We've absolutely done that. Just use a watering can. When you start getting to larger areas, we need like backpack spray packs. Now we're talking about like what we had to do in scale-up cases. Imagine this, 6,000 gallon water trucks with a spray attachment behind. What we do is we make an aerated compost tea or compost extract, and we have basically a pumper that then takes a pumper strip and Volturi's up this mixture and mixes it at a given dilution rate in with the water in the pumper truck, and we spray it on an industrial scale, right? Imagine taking 2,000 gallons of aerated compost tea, putting it on a barge, going down the French Broad River, pulling water from the river, mixing it with the aerated compost tea, and spraying the banks. Okay? So when I say I'm an engineer, so I'm thinking about how do we scale this up, right? Right? There's some things with drones that we could possibly do, okay? But we start getting into some pretty big drones with the lift capability to do this, right? So what we're looking at is like the fact that we can figure out ways and to do this. So you got 2,000 gallon, 4,000 gallon, 6,000 gallon water tankers running around here, wetting down roads, right? It's like, let's put some aerated compost tea into that as it goes down and start breaking down the volatile organic compounds everybody's breathing in, right? That's where we're going, right? So that's a compost extract. And of course,
Can I make one suggestion?
yes.
I've done a lot of that
Yep.
on smaller scale.
Mhm.
But I just wanted to suggest you can use a 5-gallon strainer, paint strainer,
Yes.
nylon bag that you can get at Sherwin-Williams or Lowe's for.
When
The
we,
problem is you
It works
can't,
just
if
as
you go
well.
smaller than 400. If you
I'm
go
running
smaller,
through backpack sprayers, garden
if you go
sprayers.
smaller than 400 microns,
It's way smaller.
the problem is if you go smaller than 400 microns, your beneficial stuff isn't gonna make it through. So
You
that's
get
why
less.
you don't want smaller than them.
when you get smaller than 400
So
microns,
filter paint, not water.
well, what happens is the fungal hyphae that we're running out, they tend to hang on much smaller than 400 microns. Okay, we'll get the bacteria through, because most of the bacteria are like 1 micron,
Yeah, sure.
2 microns, few microns, right? The bacteria make it through. The fungal hyphae, those, especially when you have a really good fungal hyphae,
Yeah,
those long strains of fungal hyphae, they'll get hung.
yeah,
So that's why we tend to use the 400 micron, and
yeah.
you get really good transfer, okay?
Alan, also, wash your bag immediately.
Yeah. All right, so aerated compost tea. So I know Oakland's probably waving at me going, go faster.
I have a quick question about this.
Yes.
Is there a certain ratio you're looking for compost to water?
Yes.
Or does it depend how rich it is?
So I was going to come back to that later, but let me answer it right now. Here's what we've ended up having to do. We figure out what we call the wetting rate of application is in gallons per acre. It's like different soils require a different amount of water per acre to wet. Like highly silt, like a lot of silt, it wets different than like clay, right? Wet. So what we do is we figure out how many gallons per acre do we need to wet, right? And we figure out how many gallons per acre do we need to do for the application of whatever we're applying, right? It's like if you're using an aerated compost tea or compost extract, we're like, we calculate for what we want to do, we want to use, say, 5 gallons of aerated compost tea per acre. We'll just make up a number, right? That might be a heavy, fit your application. All right, then what we say is, well, it also takes, we're actually having to take, in some cases, we use 1,000 gallons of water per acre to wet. There's some very resistant stuff we've had to go that high in order to get it wetted. What you end up doing is now you've got your 2 numbers. I need to put down, this many hundreds of gallons of water per acre, and then I'm going to dilute this many gallons of tea into it. And what that means is that when you put it on and it gets there, that you don't just get this mist sitting on top that then the sun bakes off, right? We have enough that it can actually soak in and the life can grab a hold, right? So I wish there was an exact number I could give you. So it's always this ratio, but it does depend upon your soil type. Does that make sense to gardeners? Yeah, right? Yeah, and other conditions in your soil. Yes.
Can you get too concentrated, over concentrated?
Hard to. It's hard to.
So if you're
Practically speaking.
You've got permaculture going on.
Yeah.
Capturing that, but you've got filtration coming through, it's dripping through into a separate container. Is that?
So
That's not too concentrated?
So I have to think about that for a little bit. What I would say is there's a whole bunch of stuff about how to do the spray application I don't have time for right now. When we get into people wanting to do this, we got people who've been working on this. We'll talk. So there's some trickinesses about how to do the actual application, how to spray it, so that it doesn't clog up, all that sort of stuff. Anybody who gets to the point of actually doing it on the ground, we'll help you out, okay? Because there's some trickinesses. I don't have time. It'll be another hour here, all the little fine details, okay? So let me roll on forward. The aerated compost tea basically is, here's the thing. I've looked at a lot of aerated compost teas under the microscope, and a lot of people who think they're making aerated compost teas, it's not quite what they were hoping. I personally will not put down an aerated compost tea. I've not looked at it under the microscope. Because if you let it go anaerobic for more than 20 minutes, the whole thing can be ruined. And that's darn easy to do. Okay? You can overfeed it. The party gets going a little too hard. And next thing you know, it all goes anaerobic. And in 20 minutes of anaerobic conditions, you can start to get multiplication of anaerobic pathogenic organisms. So it's very specific. And so what we need to do is we need to identify the experts in the area that know how to generate super high quality compost. We can look at the microscope. We can say, this is awesome. And we need to be able to take and look at the area compost T under the microscope and go, yep, this is doing the stuff. Okay? That's a 1,000 gallon brewer on the right. I mean, you could be brewing 2,000 gallons of area compost tea here. The community get together, get a 2,000 gallon brewer, be brewing this stuff up at a central location, have a schedule for it to go out in different directions and to be applied at scale. I mean, that's the sort of thing that you would do to scale up to this level of a problem, right? Okay, let's talk about biochar. I know I'm behind, but I'll keep on rolling, okay? Everybody familiar with biochar? Okay. Biochar has two really amazing things to it that we use in bioremediation. Number one, of course, is it's highly absorptive of a lot of these toxic materials. It'll grab a hold and hold on to them. But number two, it's sort of like the magic condominium for the microbes we like. You put the two things together, it changes the CEC of the soil almost instantly. I mean, there's just so many things it does, right? And so what we oftentimes see is we're using biochar in combination with things like in filter stops, we're using biochar, grab a hold of stuff. When we get back to some practical things we could do here, biochar at scale here is an option. And I'll show you something. Okay. On the left is a commercial kiln. And I know that there's some people who are going to be able to say, well, sure, we can, Alan, what is the exact pyrolysis conditions we need to optimize for this application? And I've got some chemical engineers that will tell me that all day long. They'll come in and say, you need final combustion temperature of 650 degrees Fahrenheit, blah, 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 these burning conditions and so forth, right? Well, of course, these bloody kilns are expensive and they're hard to scale up. How about the thing on the right? This is called a burn ring. Anybody use these? Okay. The thing is, are the burning conditions quite as precise? No. Can you bloody make a lot of biochar? Yes. As a matter of fact, how about this? You get a bunch of these things, you take them out right where you need the biochar, you take all this downed wood you have, throw it in the burn ring and make the biochar, quench it with the compost extract. All you need, okay, all you need is you need a 5-gallon bucket of compost, and you need a burn ring. You can go out there, and the next thing you know, you got biochar that's inoculated with good biology. You crush it down roughly. You don't even have to get it down to like, I know, the next question is like, well, okay, what grain size do we need? It's like, I don't care. We got so much to do, right, that it's like, I don't care if the burn is a little bit off. It's going to do a lot of good. Sure, we could get 12% more efficiency out of it if we did it in a blah, blah, $100,000 kiln. Okay? Forget that. Let's use burn rings out at the site of application, take aerated compost tea, inoculate it, because that way you can actually make the extract, the compost extract. We can make the compost extract right there. We can get that and put it down. Next thing you do, by the way, is you bring in chipper shredders right after it. Take all the rest of that wood, chipper shredder it, and mulch over the whole thing, right? So now, and then we come back a little bit later with aerated compost tea and spray it over the top. Okay? What did we just do? I'm giving away some of my stuff in the very end because we're kind of moving along full speed, right? Then what we do is, who's used hydro sprayers? Hydro seeders. We use a hydro seeder? Seen them do that? We come through next spring, as soon as we get to the point where we can plant the seed, we hydro seed in the proper phyto seed mix with inoculum. Okay, that's the sort of stuff we can do, and it can be done at a very large scale, and for not that much. Okay, if we just go at it the right way, very cost effective, right? We're trying to keep it down to like a couple dollars per acre. Not like, I mean, look, probably what you figured out is that the Institute I'm running is a nonprofit. I'm not trying to sell anything. I don't have a product to sell you, sorry. What I'm trying to do, so I don't have like a $3,000 per acre solution for you. We as a community need to come together and get this done.
Yeah.
We need a couple dollars per acre, not $3,000 per acre, right? Okay, so I'm trying to give you solutions that are that practical, okay? All right, let me get going faster. Let's talk about in situ remediation versus ex situ remediation. In situ means we're just going to remediate where it is. And when you get this big, you better be doing an awful lot of in situ remediation because you're not going to take all of it and put it somewhere else, right? Bloody Army Corps of Engineers wanted to move, is trying to move all of the toxic ash from the building footprints of all the burned buildings in Lahaina and put it in a landfill. They estimated 500,000 cubic yards of toxic ash.
Too toxic.
Yeah, right. And they didn't understand why we wanted to actually treat it with aerated compost tea before they moved it. They said, what good is that going to do? Yeah. Ex situ happens when we have to move it before we remediate. If it's like, I know that like in downtown Marshall, there was like, how many inches of mud on the roads? Anybody? About how much? 3 feet? Right, okay, so we're moving that, right? That's ex situ. We're going to bring that in, we're going to move it, but what we need to do is put it somewhere we can now treat it. Because that was anybody who was on top of that, I got some stories. That was some not nice stuff, right? So that's ex situ. We're going to move it. But the vast majority of what we need to do is in situ remediation. Okay. The other thing I want to point out is what we call point source versus non-point source contamination. Point source is coming from one place, like a PVC factory. Okay. Or like the picture down here is broken pipes to a septic tank. It's a point source. All the pollution is coming from one little place you can point out and go, that's where it came from. Non-point source is like the most common is different types of ag runoff, agricultural runoff. Especially where you've got like NPK fertilizer, excess nutrient coming off, but you've also got like pesticides, herbicides, fungicides that are running off. Those are all things that when we get stormwater coming across ag fields and dragging all this stuff down into it, that's called non-point source, okay? Just so you know that, there's actually reasons that we want to know the difference between those, but I won't get into it right now. But later, some of you might have to get into that. Like, let's figure out what you got, where it came from, okay? All right, let's talk about the broad ways to address contamination. And so just think about it. First is what I call physical remediation. This is physically removing debris and contaminants from soil and water. And this is after we've done large debris removal, right? We've come in, we've sorted out into our different piles that we need to have to dispose of stuff. But what we can have is we still have physical contaminants, and we need to get rid of that. What you have here on the left is an electric garden sieve. Anybody use these? Oh, wow, okay. These are super cool. Let me, and you can get them really big. What you have here is you have this barrel and it rotates, and you load in contaminated soil here. And what happens is these holes are small enough that the soil falls out here, and all of the debris comes down and falls here. So you just shovel in, and you get little pieces of metal, little pieces of plastic, everything else comes falling out the end. And so what we do is we can do this for like high value places. You got a garden, you're like, I don't want all that debris in my garden. You can go through the topsoil and you can, it's a pain. But this is what we would call, physical remediation of soil. We're gonna get out all the chunks of stuff, right? Now, what you have on the right is what's called the Baltimore River Trash Skimmer, solar powered, and what it's doing is it's skimming floating trash off of the water, right? And so it just goes up and down and it gets all that. So there's a bunch of different ways. I just want to let you know there's a lot of different ways of doing this. And I call it physical remediation because all we're doing is we are getting the physical debris out. It's not like the stuff this big that we can haul out. It's the stuff that's small enough, right? Especially plastics, okay? That's called physical remediation. All right, decomposition. We chemically break it down. I have fungal hyphae there. Everybody recognize that? Fungal hyphae. They're really good at it. There's bacteria that are good at it. By the way, floating islands. This is one of the ways we do, we basically do it in water. Decomposition in water. What happens is, that's a picture of it, and I'm going to take you to a cross section. What happens is the roots actually go down into the water and biofilms get established on the roots. And basically the good biology we're talking about, they live in biofilms on the roots and they start to filter the water as it goes past. This is a very effective way of large-scale water bioremediation using decomposition. Okay? So that would be something that could be looked at in this area. Okay? I know I'm going fast, but I'll give you some. Bioaccumulation and extraction. This is oftentimes used for heavy metal contamination. We get specific plants that are really good at bioaccumulating like a particular metal. Okay? And then we let it grow, and then we actually harvest that plant and take it, and oftentimes we then burn it under controlled conditions, collect the ash, and that becomes toxic waste we have to get rid of, because unfortunately elemental stuff like lead and cadmium and hexavalent chromium and all that kind of stuff, you can't break it down. Okay, we just got to bioaccumulate it and extract it and get it out of the environment. That's what we call bioaccumulate and extract. Okay, immobilization is another one. One of my first in-person PDC was with Jeff Lawton, by the way, and he called it putting it in carbon jail. Yeah. What happens is a lot of the bacteria and the fungi, as they're building soil organic matter, they'll find the nasty stuff that they don't like, and they'll say, "Oh, we don't like that." And they'll actually take that and they'll build it, the soil organic matrix around it, and it becomes chemically inert as long as that soil organic matter is there. It's a form of chemical immobilization. How many of you have heard about Jeff's Green the Desert thing? Right? You realize what he did? That was salted hardpan. And like, where did all the salt go? Well, it turned out that the microbes didn't like the salt, so what did they do? They locked it up in the soil organic matter. It was still there, it was just chemically inert. That's how he basically desalted that soil. Okay? And it turns out there's other chemical compounds that will, the same thing can happen. So that's called immobilization. And then oxygenation. We can, especially in water, but you can also oxygenate soil if it's compacted. Okay? Everybody use flow forms? Yeah, this is a flow form. You can actually add a couple ppm, like at least 1 ppm of dissolved oxygen for each stage coming down through here. What I want to see, for anybody who's a water scientist, what we try to do is we try to get the water at least 8 ppm dissolved oxygen. I prefer 10. Okay? We take a dissolved oxygen meter, we're going to look at it, we want to make certain that our water is oxygenated. If it is anaerobic, we've got a problem, right? We get into eutrophication, we've got a problem. So there's other methods we use for oxygenation. I've used this on a large scale. I've happened to use this particular vendor before. It's not an endorsement, but I have used them. This is Vertex bottom aeration. You'd think that the bubbles going up were the things aerated. What actually happens is it entrains a column and it creates this huge convective cycle in the lake. You can, if you do it properly, you can actually turn a lake over a couple times a day using nothing in this. You have an air compressor that sits on the shoreline and a weighted air hose that goes down, and then those things go at the bottom and they create these columns of bubbles going up. They turn over lakes. And I have reoxygenated big lakes that way, okay? So if you run into eutrophication and you run into situations where you got ponds and lakes that are going anaerobic because of excess nutrient runoff and so forth, there you go, okay? That particular one is from a company called Vertex. See the little source down there? Vertex aeration systems. He said, not a particular endorsement, there's different companies that do it. I just happen to have used them for a couple of things before, but there's other companies that do it. And then competitive exclusion. This is one that's not talked about in the books very much. It's something that I've done a lot of work with. Competitive exclusion is an idea in ecology which says that it's really hard for two organisms to be in the same ecological niche because they're competing for the same resource, right? So what if we got all the good guy microbes in there, right? And they claim the territory. What that's going to do is keep the bad guys out. And when we start talking about buildings and making certain that buildings have good microbiomes, one of the things I do is competitive exclusion. Notice I have a lactoferment. Little secret. Whenever I build a new building that I want to be ecologically sound, before I let them actually use the kitchen, we bring in sauerkraut and milk kefir and let it run for a couple of weeks. What it does is it gets lactic acid bacteria. And what it turns out that we know that lactic acid bacteria will actually competitively exclude a lot of molds. Okay? Yeah. Now, we haven't got the science yet on whether how much it will hit black molds. I know I'm going to run over, but do I have your permission to go a little bit longer?
Yes.
Okay. So here's the thing. How many of you have houses that had some flood water in it? How many of you had those houses with flood water in it with drywall? Okay. Architect we were working with was looking at building biology and this problem of black mold in buildings. And she started looking at drywall. And she started to wonder, why is it every time, you notice that anytime you just like look at drywall and think about moisture, it creates black mold. What she found out is that of all the samples that she looked at, 90% of them, the paper backing had black mold spores in the paper backing. Okay? Now gypsum is kind of like black mold food, right? So what your drywall is, commercially available drywall, is I call it black mold chia pet, just add water. Okay?
That's crazy.
It is. You wonder why I don't build buildings with drywall anymore.
No, I wouldn't.
Okay?
I wouldn't wonder.
So this is why every time you get water into a house that has drywall in it, that you have a black mold problem. Okay? It's because you've been living with dormant black mold spores all around you the whole time. All you're just waiting for was moisture. Okay? So anyway, that's something that competitive exclusion really has a hard time stopping, but because it's just everywhere, right? So, but competitive exclusion, another method we can use that, okay? All right, let's talk about the types of contamination that you're probably looking at. And I break them into 4 broad categories: physical, chemical, biological, and radiological. So physical is your trash and your debris, everything from very large, like, there's a van that's been like just taken away by flood waters and run into a tree, right? I'm sure everybody's seen a few of those, right? Okay, all the way down to tiny, almost microscopic particles. Chemicals, of course, the problem with a lot of the chemicals is they can dissolve in the water, and now you cannot physically filter them out. You have to actually get rid of them in another way. You got to do different kinds of absorptive filtering in order to do that. Biological is bad microbes that are harmful or that produce toxins. Why is black mold bad? Well, it can produce compounds that a lot of people are allergic to, can cause respiratory distress and so forth, right? So, that's biological, and then of course radiological, radioactive materials. Okay. So physical pollution. Physical pollution, I'll break that into debris, which is man-made objects of various sizes that end up as litter in the environment. We got mesoplastics, which are, and by the way, these particular terms, different people have different ideas about what exact size range, but these terms have now become standardized and people will argue about the exact size. I'm not bothered with it. The whole idea is mesoplastics are these little bits of plastic. You've probably seen them and you pick them up, they're little snippets of plastic like that. Those are mesoplastics, right? Microplastics are tiny fragments of plastics that are about 20 micrometers up to 5 millimeters, and then nanoplastics. These are nasty. And we don't hear about them. You hear about microplastics? Yes. How many of you have heard of nanoplastics? Mm-hmm. Cool. That's good. Nanoplastics. These are 1 to 1,000 nanometers. They're so small they can penetrate into tissues and cells. And so we wind up with nanoplastics inside of our cells. right? The whole thing is, guess what? Macroplastics become mesoplastics, mesoplastics become microplastics, microplastics become nanoplastics. That's the process. That's where you get stuff like photo deterioration and so forth. They break apart because they've been illuminated by ultraviolet light from the sun. So we won't get into all of that, but that's your physical stuff that you're dealing with. Okay? All right. Next, we've got VOCs and SVOCs. Volatile organic compounds and semi-volatile organic compounds. A class of chemicals that can easily turn into a vapor at normal temperatures, making it possible to inhale them easily. Short-term exposure can create respiratory distress, while prolonged exposure can result in long-term and chronic health conditions. Common sources include fuels, solvents, hydraulic fluid, cleaning agent, and degreasing agents such as TCE, which is trichloroethylene. And by the way, there's some of these things that you breathe them in for very long, you can have permanent lung damage. So it's like, don't play around with them. I know there's why people are like, I'm gonna tough it out. Toughing it out isn't worth the rest of your life having respiratory illness. So we're gonna talk about PPE in just a minute, okay? But those are your VOCs and your SVOCs. What you typically find is that they're most intense at the very beginning, and then they begin to dissipate over time. Next one, persistent organic pollutants. Okay, class of organic compounds that do not break down easily in the environment, therefore causing long-term pollution. Examples include dioxins and furans, PCBs, certain pharmaceuticals, and certain classes of pesticides, herbicides, and fungicides. Many of them are halogenated organic compounds that are fat-soluble, technically lipid-soluble, okay? Meaning they can potentially be bioaccumulated up the food chain. Exposure to POPs is associated with a number of chronic illnesses. I didn't have space here to list them all. Okay? Yes, I put glyphosate as the fun one. People are like, but Alan, glyphosate breaks down in the environment. How many of you think that's true? Okay? We found glyphosate in certain soils after 20 years, okay? So they don't believe you when they tell you that they break down quickly. Okay? By the way, here is, when we get into the details of it, here is some research done by a gentleman out of Arkansas, where what he has is he lists out these different plant species, and then here are the particular pesticides that they tend to break down really well. So when we get into it, there's this kind of detail we can pull out. I don't have time to go through and say, if you've got atrazine, then this is what it is. We can give you all that information, okay? We just know that this kind of information is out there. And by the way, some of the stuff coming out of the Ozarks is a climate analog for here. Some of these plants will work here as well. So these particular tables probably pretty useful. Okay, we can pull these things. Okay, PAHs, polycyclic aromatic hydrocarbons. They're called polycyclics because they've got multiple rings of hydrocarbons. I'm not going to get into these too much, but they usually come from incomplete combustion of fuels, petroleum and biofuels. Okay, so you're probably not going to see as many of them here because that wasn't the kind of event you had, so I'm just going to go on past that. PFAS compounds. These are the thing that people ask me, how do we bioremediate PFAS compounds? My answer is, the answer to PFAS compounds is do not make them in the first place. I asked one of the world's best PFAS chemists how to break it down, and she laughed at me. Right? She said it's an industrial process. We're still trying to find some way to bioremediate PFAS. It's like the problem isn't the bonds in the PFAS compounds. Most of the reason that we can break down most organic compounds is because life can get a little bit of a bonus of energy by eating it. Right? PFAS compounds, some of these bonds are so bloody strong that it's like convincing any of the living organisms to try to go after them and break them down. We're probably starting to see it, but we don't have a great answer for it yet. Yes?
Would this be a good candidate for something like immobilization? Do we know if anything is immobilizing?
That's a great question. I wish we had more science on it. There's so much, I don't know, do we have any research scientists in the room? Okay, I mean, I need research scientists because what we need to be doing is that research, right? It's like I need PhDs and grad students. We can document all this stuff and get it out there in the literature so that when we go in and talk to the people, that we have the science in peer-reviewed form. That's what I really need, right? So that's a great question. All right, let's keep on going. Heavy metals. I think everybody probably knows about heavy metals, right? These are typically bioaccumulate and extract. That's typically how we go after them. Okay, and then of course, here is a list of plant species that will preferentially bioaccumulate. Now there's multiple pages of this, I just gave you one example, right? Like here's like, oh, I got chromium, what do I want? Well, let's use brown mustard or willow or birch or whatever. This is how we can, these are, what we find out is that there are certain species that preferentially bioaccumulate and extract metals. Biological contamination. Okay, this is bad biology. I tend to go after this with competitive exclusion. Okay, this is a good way of going after it. Things like fecal coliforms and E. coli are common in water after flooding. Yeah, and this is where we were taking and doing E. coli cultures in a agar medium in order to see how many cultures of E. coli we had in the water sample. Okay. Excess nutrients, of course, is often something that we see from ag runoff. This can cause eutrophication of your waterways. I don't know how much. Anybody have an idea of whether you think you had much of that happening here?
Yeah.
Yes. Okay. So one of the things, if that's the case, you might have especially nitrogen, phosphorus, potassium runoff. You may be seeing eutrophication in especially water impoundments, ponds, lakes, so on and so forth. You want to look at that. You want to watch for algal blooms, which of course is a classic sign of excess nutrient. And you want to look at dissolved oxygen and see if it's eutrifying. If so, we want to do phytoremediation, and we want to do, which is floating island. Oftentimes we want to oxygenate. I can give you a lot more strategies for oxygenating if you need it, okay? And then we've got radiological contamination. Now, we don't know yet exactly whether, we've heard different stories. You might have some radiological contamination along. I've looked at a couple things that I've run around. This is a Geiger counter. I brought my Geiger with me. We've done some looking as I've run around. I haven't found hot spots yet, which is good. Okay. But we've got a couple people around that now have Geigers. If you think you might have an area that's contaminated, then get in contact. We'll get somebody through to come and take a Geiger counter and see if you got anything, okay? But this is, one of my letters in my letter soup I didn't put on there is I am an RSO, Radiation Safety Officer, I'm a certified RSO. So I've had to know how to handle radioactive waste and everything, okay? And so, yeah, we know that there's potential for radiologicals to have gotten loose during some of the stuff that happened, especially up over towards Oak Ridge, right? So we'll take a look at that. Okay, so Oakland, you'll come on down.
What would be some signs of radiological contamination in a particular area?
So the question is, what are the signs? The problem is that a lot of times you can have hot radiologicals, and unless it's hot enough to make you have symptoms of radiation sickness, it'll be a long-term thing. So best, if you think you might have it, to just do a radiological survey and see what your background radiation is. Okay, so go ahead and get suited up here. You got gloves there. Has anybody seen the PPE one sheet that we put out? Okay, we sent it out, and I know that they've been using it around Marshall. What we did is it's a front and back. It shows you basically this, with a lot of more information on it, okay? Physical protection on the left and chemical protection on the right. Yes. What? Oakland's going to put her gloves on. She's only going to put one layer of glove. It should be two. Okay? When you are working in something like the mud that was in Marshall, this is what you should be wearing. Okay? This is what we call hardware store PPE. The reason is that we intentionally got all of this from a Home Depot. Okay? Every bit of this is available at a Home Depot. And what we have here is we have a Tyvek. There's a spot called Workplace Safety. And Oakland, if you'll come over this way just a little bit so they can make certain they get you on the live stream. Okay. This is keeping all of the skin from being directly contacted. We'll use 2 layers of glove, typically an inner nitrile and an outer work glove. Okay. This is a respirator versus an N95 mask. Here's the deal. An N95 mask will help keep dust out. It will not stop volatile organic compounds. You breathe it right through the mask. So if you're on top of anything that's got volatile organic compounds or semi-volatile organic compounds or certain PAHs, then you need, this is a P100 respirator, which is what I recommend. Okay, she's got safety goggles on. This keeps all that contamination out of her eyes and off the mucosa of her eyes. Okay, and there is on the back of this one sheet, and you will actually, and if you need to, you'll don rubber boots over top of this. Okay, big rubber boots. I was told that in Marshall we had some rubber boots turning green from contact. So the one sheet on the back will show you how to take this off safely, because once it's contaminated, if you don't take it off safely, you'll just get it on you. There's a particular order of taking it off. The back of the one sheet will tell you how to take it off, yes.
At this point in time, do we know what parts of town we need this whole equipment for?
That's a great question.
Commercials, I guess, but like people speeding up the French Broad or Carrier Park or.
I would say if you are downstream on the French Broad from the industrial sections of Asheville, I would be very careful. We walked over it a little bit yesterday. We didn't have full, but I could feel it. I had a little small, not full respirator on because we were dealing with some of the local people there. Wonderful people we're sitting there talking to, and they're walking around with no PPE and so on and so forth. You got kids down there playing and so on. But, you know, I know we've got people who have wound up in the hospital from being exposed to this stuff. Okay? So I wanted you to physically see what it looks like, right? In some of this stuff. By the way, I had to work on some sites where this isn't enough. You go to full hazmat suits. Okay? You'll notice on my little one sheet it says this is what you wear in areas of moderate contamination. Heavy contamination, we go to full hazmat suits. That is, if you don't want to get sick and wind up in the hospital, that's where you would go. Thank you, Oakland. Yes. Yes.
So a lot of them volatilize out after, depending on a week, a couple weeks. We're going on a month.
Yes.
Do we need to be concerned about VOCs that are maybe at depth? Something that's
So-
buried, or are those also well?
Yeah, so here's the thing. Typically what happens is if you've got contamination down deep in the soil, you'll get a lot of volatilization out of the first couple of inches, and then the volatilization will slow down. If you disturb that soil and you've got contamination down deep, as soon as you turn it up, guess what? You just expose a lot more of it now. Boom. So yeah, at that point, if you have to just like move it or disturb it and turn it over, then you need to be very careful because you might expose stuff that's down deep. All right? So we will give, there's this sheet, some of the people that are the Lahaina team helped me pull this together, and it's available. What it's done is it's set up as a two-sided one sheet, and you can take it and laminate it, and when you're working with volunteers, you can hand it to them for training, and it'll show them how to put it on and how to take it off, and talk about some stuff from there, right? So, good. I can see that the two things. One is that there seems to be still a lot of interest. Number two has been sitting for a long time. So, and I know we're going to run a little over because we just got so much information. So, here's what I do when I'm teaching. And Oakland's been in a lot of my classes where I teach like 4 1.5 hour sessions in a day. Okay? Yeah, and so what we do is, like I said, everybody feel free to stand up for just a moment and stretch. We're gonna take a 30-second stretch break and then sit right back down. Sort of self-care because we've been sitting for too long.
Stick this in here.
All right, so WNC. Please, and we'll go ahead and on floor. Okay. So we're going to find out how many of you are really serious compost makers. Okay. What is this? Yeah, it's a Riotemp compost thermometer. This is a 4-footer, right? So who all does windrow composting big enough that you need a 4-foot Riotemp? Anybody? Seriously interested? Yeah. Because that's the size we're talking about. I've been talking to some of the folks here. We're talking about moving over to windrow composting to have large enough scale to be able to do it. It's like this is what you're talking about. We're talking about windrows, and you use this probe because the actual probe is there, and then you read it here, and you take this. For those of you who haven't seen this before, you take this and you drive it in the middle of the windrow, and it lets you read the temperature at the center, so you can manage your compost pile. Okay. So I brought this. This is one of my Riotemps that we use for like large scale production. And you transport it inside of this PVC to keep the probe tip from being damaged. All right, we are getting close to the end of the presentation. I want to talk about emergency response frameworks really quickly, and then we're going to get into Q&A and go forward, okay? So here we go. First steps in an emergency response. Now, I'm starting to try to define this as what we call an emergency response framework, because I've noticed that there are a lot of folks who have been doing really nice things with bioremediation, but it's usually, a little bit here, a little bit there, and it's like, well, when we have something like Lahaina or what just happened here, we need a full response framework, okay? And so like I said, I've been doing systems engineering. They usually bring me in a situation where like, this is a big mess of a problem, and we need to figure out the whole system response to it. Because that's kind of like the way I like to think, and I've got, three and a half decades of experience of thinking that way, and having to try to solve problems at a big scale. So, okay, what does it look like? Well, the first thing we should do when we come into any area with an emergency like this, step one, these are the first steps in an emergency response, identify potential point sources and non-point sources of contamination. What we should do is we should immediately crowdfund, okay, everybody tell me what you know of. And somebody says, oh, I know about this PVC plant. Okay, and I need to know about every industrial source. I need to know, okay, we've got big ag over here with non-point source, runoff, so on and so forth. We want to do that. Next thing we want to do is we're going to research what specific contaminants that might have been introduced from those sources. And the third thing we want to do is we want our GIS experts to map all of it for us. Okay? So when I do any kind of large-scale response, the first thing we want to do is get the GIS team going. Okay? I'm sure we got some GIS experts in the area. Okay? I think we have a volunteer. Okay? Just out here is GIS, like very experienced GIS people. 1, 2, 3. I got it. 4. 4? Okay. We got at least 4, right? Okay. So you'll immediately see what I'm up to. What I want to do is I want to map all my point source and non-point source. Then what I do is I want to run a stream channel network analysis on the topography, right? And then what we can do is we can take those things and we can look down the watersheds and see where they probably went. So then we can kind of know, hey, this particular contaminant, source of contamination probably went here. Other than that, we're like, how in the world do we know what to test for? Because this turns out to be the case. Testing for everything is bloody expensive. Now, oh boy, I can get into like 3 hours of discussion on testing, right? But it turns out to be bloody expensive. Everything you need to test for typically costs you more money. We need to be able to take soil. So the next thing we do is we take samples of soil and water and send it out for testing. Okay, now the question from hell, which labs do we trust? Okay. There are some labs I just do not trust. Because we'll send them stuff that we know is contaminated with a particular thing. They'll come back and say, no, it's fine. Right? So there it is. But what we need to do is we need to know what our highest priorities are to test for so that we don't have to pay. It's like, you know, I was looking at some stuff for well sampling testing. The test I wanted to run on a couple, what I wanted to do is like look at it, I'll get into some water responses. It's like, we can get hydrological maps, map out where we know where the aquifers are. I'm like, what we really need to do is go to a couple of wells pulling out of that aquifer. We need to take samples from that, and we need to get every bloody test we can on that well. That's a $3,000 test. It tests for 365 different contaminants. Okay, and just see if there's anything that has actually gotten from the surface water down into the groundwater. Okay, and then if we find it in that particular aquifer, we're like, oh, well, maybe every other well pulling out of this aquifer needs to look for that. And what we can do is we can say on the GIS map, this whole area, if you go to a well at this depth, it's probably pulling out of this aquifer. We can then get all the well permits. We can overlay it as a GIS layer, and we can come up and say, all these wells are pulling probably at this depth, they're pulling out of that aquifer, and then we can start to go in and be able to tell people, hey, your well is in this aquifer, we've tested for this, we found this contaminant, we recommend that you pay for your well to get this special test to see if you're seeing it, right? Because we can't have everybody pay $3,000 to test a well. It's crazy, right? So we need a strategy. It's like, let's raise a little money, let's test critical wells in certain places and see what we're seeing, right? And then maybe test them again in 6 months and see if we're seeing anything show up from surface water incursion into the groundwater, right? These are the kind of things we need to be doing, okay? And that allows for us to take and find out what contaminants we're really dealing with and then specialize with testing so that you can go into people and say, in your area, probably looking at this, test for this. And then that's one special test that costs a few dollars, not test for all these things, hundreds of things, and it costs you thousands of dollars, okay? That's the strategy we use. Okay, start emergency response bioremediation steps, focusing on breaking down VOCs and other high-risk contaminants. Aerated compost tea sprays can be used on highly polluted soils to jumpstart the decomposition process. It's like, to me, if I could do it perfectly, before I let anybody get in and start taking off heavy debris, we just go through with aerated compost tea, dilute it, and spray it over everything and let it sit for a week. Because now the risk of being in there goes down dramatically. Okay? Then we go down and clear the debris. Then we go through and start doing physical remediation, so on and so forth. That would be the ideal. Okay? By the way, what you have here is, this is one of Chase's maps. And what we have is he's just pulled, you can't see all the detail, but what he's done is he's actually categorized the sub-watersheds. And you can see the stream channel network. And so if we put a point source on here, we can actually follow it down the watershed and see exactly where it would go. Okay? So we can also see which areas of the land are nested watersheds inside of inside of larger watersheds and so forth. That's the sort of stuff we need to do. We start needing to map it out and figure out where all these contaminants probably went. Okay? Making sense? Yeah. All right. How about soil remediation phase 1? What do we need to do? Well, we talked about this already. Okay? Remove large debris and then do finer remediation, which is like your sieving and so forth on high priority areas. Like if it's going to be a garden, if it's going to be something that's like that, we do that. Use burn rings to produce biochar from downed wood, quench it with compost extract, break it down roughly and apply to the soil. Use an industrial chipper shredder to turn the downed wood into mulch and apply it all over the biochar, spray diluted aerated compost tea over it, and then come back into the spring and hydroseed it. Now, here's the thing. In the meanwhile, the test results come back and it tells us exactly what we're dealing with. Now we know which plants we want to put into the hydroseed mix in the soil. Okay? That would be your phase 1 of broad soil remediation. We should be able to do that at scale. Right? Hydroseeding, we can do thousands of acres with big hydroseeders. This is, by the way, what one of the bigger industrial hydroseeders looks like right here. Ever seen these things on the side of the road? Yeah, that. We just repurpose it. What we do is we put our seed mix in it. We inoculate those seeds. We put aerated compost tea in it. So we're putting down seeds and inoculum at the same time. And then you start getting it, and we get a cover crop coming in. And it's typically about an 8-special mix I like to use. Okay, so we're usually around that. So, and I won't get into it, but that's covered.
Riparian blend?
Yes, so what we can do is we can do a riparian blend if we're going to be spraying into riparian zones. So it can be customized down. So you can come right back on with those barges and spray spray off the barges after, It's like that's what we need to be doing. But we need to know exactly what we're dealing with. Are we having to do any bioaccumulate and extract? If so, then okay, we've got to think about that. Right? Okay, water remediation. I'll hit these super fast. The last two, then we're going to kind of be done. Use biofilter socks to keep additional pollutants from getting into the waterways. Those are those socks with the mycelium and the biochar in them. Remove large debris and then use filtering and skimming to clean up smaller debris. Assess water bodies for excessive nutrient runoff and eutrophication. Use oxygenation strategies where needed. Consider floating islands where chemical contamination needs to be addressed. And then the additional steps that we need to do from there are highly dependent on the exact contaminants that are present. But then hopefully we can get the test results back from the water samples. We know what we're dealing with. We can come up with very, very customized remediations. Last thing I'm going to hit is building remediation overview. Okay? And boy, this is its own class. I'm just going to say this. Check all buildings for structural damage before entering and after any event. Do not enter buildings that are structurally unstable. That's pretty obvious, right? Assess buildings for possible non-remediatable chemical contamination that might have been absorbed by permeable building materials. The news is that there are some permeable building materials and certain contaminants that once they get in the building, I don't know how to get them out of there. They've absorbed into permeable stuff. And if you get something nasty enough, probably the best thing you can do is to take that building down and replace it. I'm sorry to say. Okay? So the first thing I want to know is the building structurally sound? If so, did it get some chemicals in it I can't fix? If the answer to either one of those is if it's unstable or it's got contamination I can't fix, then maybe it's like, I don't know how to fix that building, maybe replace it. Okay. But if that isn't the case, what we come in is we do what everybody said to do, right? Pull out all the drywall, pull out all the water damage stuff. Contractors know how to do that, right? They know how to go in, pull out drywall, carpet, subflooring that's been damaged and so forth, strip it down to the studs, and let the whole thing dry out, air out. And then they know how to rebuild it. What I'm going to tell you is I think that once you go through a rebuild, you got to biologically inoculate that building to get competitive exclusion going so that anything that was bad biology that tried to get set in during all that probably isn't going to hit you anymore, okay? So that's kind of a super fast running overview of that. We can talk more about that if we need to in the future, okay? So last thing I'll say is I have, this book is the one I would recommend. I talked to Lila the last Thursday about some of the approaches I'm talking about here. She's very highly experienced in this area, and this book is amazing. As you can see, it says a grassroots guide to healing toxic and damaged landscapes. It is designed for groups like us to come in and fix it and not have to go off, because there are companies that will try to come in and charge you huge bucks. In a few cases, they're worth it because there are some specialized things that require very special, like groundwater. If you get groundwater contamination, that's hard to fix. okay, things like that. But a lot of the stuff we're running into, we can fix, okay? So that would be the book that I would recommend right there, or Repair.
Yeah.
Yes, yes,
Did you bioinoculate buildings before putting new materials on?
yeah. I would do it before and after. Yeah,
Are you
yes.
talking about using your compost tea and extract in order to spray buildings down?
No, what I find really interesting is that the labs, the lactic acid bacteria, seem to be really, there's a lot of research in food that the labs actually keep off the molds. So I'm really interested in that, because what we know is when we take and like do sauerkraut in a building, what you start finding is you can take samples off of like every surface in the kitchen, and guess what you find under the microscope? You find labs. You find lactic acid bacteria and other bacteria out of that sauerkraut and so forth. And then the same thing you get when you do milk kefir. The reason I use milk kefir is it's very biodiverse, right? It's more biodiverse than water kefir, and it's complementary to the sauerkraut. So I tend to go in with both milk kefir and sauerkraut, or whatever lacto-ferment you happen to like is. If you like kimchi, like cabbage kimchi instead of, like, go for that, right? Okay, if you like pickles, lacto-fermented pickles, I'm talking about the real lacto-ferments, not quick pickles, right? We're talking about really lacto-fermented stuff. That's got the labs in it. That's amazing. That's what I've been using, and it's really amazing. The chef that's worked in my kitchen at my house tells me he did a game where he tried to get food to spoil in my kitchen. And he said, I have a hard time getting food to spoil here. Yeah. Just I bring in sauerkraut jars and milk kefir and let it sit. Yeah.
I've seen spray, like heater sprays.
Right. So, I mean, that just seems to work. It just gets in there, and then after a little bit, if you keep on doing it, it just gets in and it covers all the surfaces. Competitive exclusion, you're in business. Yes, ma'am.
So your sauerkraut and kefir are open to the air?
Alan,
No, I just do them normal. Yeah.
for the benefit of people online, can you repeat the question when they ask?
Oh, okay. Thank you. Yeah. So, no, I just do like a normal ferment with the. I had a picture of it with the little water lock. What happens is when the off gases, you just get beneficial bacteria going everywhere, right? When I'm doing the milk kefir, I put a coffee filter on top so that it can exchange air when we have the milk kefir out, right? That's a one-day thing. We take the kefir grains out and take the kefir off every day. So that's what I do with that. Just that. If you're already doing it, the way you normally do lacto-ferment, the way you already do milk kefir, let's just do that. That's all it takes. So we are moving into Q&A. So we're going to do this for a few minutes, and then we're going to go over to V, help introducing people that are doing different projects here in the area. So let me just kind of okay, I'm never going to get to all the questions. So let me ask you to prioritize the questions this way. I am happy to stay here after we're done tonight and answer questions. If your question is very specific, like to your circumstance, please hold that question and just come up to me afterwards and I'll do it. If it's a question that you know that everybody would love to have the answer to, let's prioritize those questions so that I can answer those and everybody can hear it, right? So let's start right here and we'll go from there.
Okay. I live in Marshall, downtown Marshall. And I've been walking by the river.
Yes.
And the issue with the river, along the edges of the river, is that it's been accumulated with sand. I mean, it's just like flush with sand.
Right. Yes.
So how would spraying that tea on that stuff be effective in those kind of conditions?
Right. So the question, let me.
The other question I have real quick is that I'm wondering, on the island, like Landon has an island, there are all those trees that were fallen. Could those then be chipped? And
Absolutely.
on the island so they're called off?
Yes, that's what you should do.
It's part of that process.
Absolutely. So here's the question. We're talking about where you basically have all this silt and, a lot of silt. I saw a lot of silt It was brought down the river, it was deposited, especially in the back eddies, like where, you know, like out of the active channel, up on the sides, you got a lot of, because you got the back eddies happening, it was dropping out a lot of the stuff it was transporting. And so now you've got, in some cases, inches to feet of silt and mud and so forth, and then a lot of it's toxic. That's exactly the stuff that you should be hitting with what we just talked about. You should be hitting it first with, you know, some sort of aerated compost tea or extract, knock it down, then, yep, biochar, some of that wood, chipper shredder some of that wood, get the biochar down there, get the mulch down there, come back and spray it a little bit later with aerated compost tea and hydroseed over it. Boom. That's what I would be doing on all those areas that are up out of the main river area that have this toxic stuff on top of it, all of it. Let's go that way, all of it. Let's see. So that answer the question? Perfect. All right. Let's see. I'm a little systematic because if I don't, I'm going to miss people. Let me start right here. Just kind of work this way across really fast. So let's come right here.
So I've been doing the reconnaissance for the past 3 weeks to a month, and I've been everywhere from Elk Mills to Burnsville to Irwin, all those different places. And I noticed that at every rural holler, they're burning debris.
Yeah.
Is there something we can do to get them to stop doing that, or is there a remediation technique for that?
I mean, it's just literally it's education. You go in there and say, you know, you're burning all this.
It's mostly Department of Transportation.
Oh, yeah. So okay. Here's the deal. Okay. If it's Department of Transportation, we just got to find the right people.
They don't speak English.
Come talk to me about that, because it's like having to deal with, well, I don't want to get into this. We'll just say that in Maui, for the Maui situation, we wound up with testifying as expert witnesses for the government of Maui was FEMA, Army Corps of Engineers, EPA, and then me and one of our mycologists. And so, yeah, I fought those battles before. Okay.
Thank you.
The thing is that you have to understand that the people, the frontline people, are not always. They basically have their standard operating procedures, and they don't have a lot of latitude sometimes. You got to find the right people with the right leverage, and you got to have the right conversations. Okay, so it can be very frustrating at times because they're just, you know, it's very, in some cases you run across individuals that are not very pleasant. In a lot of cases you run across people who would like to be helpful, they just can't. They're in the system, right? And so we have to learn how to work with the system. What we need to be doing is going in and really, it's at North Carolina Department of Transportation?
Both North Carolina and Tennessee.
Tennessee, okay. That's a little easier to work with than federal government, believe it or not. The thing is that you need, if you get the politicians at the right level to basically say, work with these people, then it happens. If you can get the county government to say, hey, you're here, you're working with us on this, we would like for you to work with this because we're trying to clean up these things, you have a possibility. If you just show up and say, hey, we want to change what you're doing, that's hard. Okay. So I'll just say that. We'll have a conversation with you. That's a whole bunch of strategy of how to work with that. But yes, I mean, all that stuff is potential, instead of burning all of it and creating more pollution, it's like, okay, if you're going to burn it at all, make biochar, and then let's chipper shredder this stuff and put it down as mulch, just remediate some of this instead of burning all of it off, right? And by the way, some of this, if the wood absorbed anything toxic, then that remediation process on those wood chips cleans that up too. So let's come here.
I'm just curious about any other biological solutions to support the wells in our area, because they're coming back with E. coli and coliform, I don't know what parts per million. So are there any ways to treat the soil, to have it infiltrated the well system, or just your comment on how to support the bio? Because chlorine also shifts the biofilm and, you know,
Yes.
causes trauma to the pipe.
Absolutely. I don't like chlorine for a lot of reasons. I will have to go into here. What I'll say is almost always when we see E. coli and fecal coliforms in the wells, it's overtopping. It's gotten contamination from the top. That's usually how that has happened. Because the only other way it could happen is for like E. coli and coliforms to be down in the aquifer and coming up. That's not very common. It happens, but it's not as common. So in those cases, there are very specific ways to go after that. Oftentimes, they want you to chlorinate and then flush. That's the way they typically want you to do it. The problem is that if you put any biology down in there, you can start to get biofilms on the pipes. So yeah, and we don't want that in well cases. So the best thing I can say is we should be designing our wells so that they are protected from flooding in the first place, right? That is, if they get flooded over, that they're locked down and they don't get flooded from the top. Okay, but you maybe, if you have significant E. coli and fecal coliform incursion into a well and it does not self-clear pretty quickly, you may actually be down to chlorinating and then flushing. Anybody else has a better solution? I don't know everything on this, by the way. There's so much to know about this stuff. We probably have other people in the room that
I have
other
a
ideas.
UV light.
Sure. So so UV sterilization, we use that and so forth. The problem is we got to actually, you can't get, I don't know, we might be able, you have to take a UV all the way down and drop it all the way down the water column to sterilize the inside pipe surface. That's your problem. And so, yeah, I've designed off-grid water systems for communities where we have them running off-grid water. And we use UV sterilization as the last phase of that whole decontam, right? You push the water by it and it goes. The problem is that you have to have very low particulate matter in the water for that to work. And we would have to actually UV the entire wellbore in order for that to really work effectively, if it's contaminated down the wellbore.
Well, you can have UV inline piping that comes from its source, from the well
Right.
before it enters into the house.
Right, and I said, we've done that, I do that all the time, but that doesn't, if we're trying to actually clean up the wellbore, it doesn't do it. But yes, I do that all the time. It's the last stage that we do in a water cleanup system. So let me go.
Chlorine dioxide tablets.
Oh boy. Let me do some more research on that one because, yeah, let me do some more research. If there's anybody who's got experience with that, this is one of the ones that unfortunately I haven't had to deal with too much of this.
I drink it.
Me too. I drink it too.
Yeah, but I mean, I'm a purist. I like to clean it up. I mean, I'm known as being very aggressive on trying to come up with green ways of doing it. I got like about 5 minutes. What time we got right now? Whoa, we are okay. I'm sorry. I'm gonna have to do this. I'm gonna take 2 more questions and then we gotta go. Because otherwise, I'm gonna be here until 10 o'clock doing this. Let's go right here and yeah.
So for pre-existing remediation practices that have
Yeah.
existed for decades
Yes.
at the industrial level,
Right.
are there any of those remediation practices that are used in the commonplace that you support, endorse, that FEMA might try to pull out, or?
So I do not know of any bioremediation strategy that FEMA has done. What we're trying to do is we're just trying to get FEMA to tolerate us doing it.
What about industry in general, like cleaning up gas stations and ground?
Yeah, so cleaning up things like petroleum products. Paul Stamets has done a lot of stuff. He's got the name recognition. He's gone in and got some governments to work with him. And so he's done some places where they've had some really good work on cleaning up certain things like petroleum and so on and so forth, right? So yeah, some really good science around it. It's very documented. That's becoming a little bit more mainstream. There's now a couple of companies that are trying to commercialize that and bring that on market as a commercial response.
Well, what has been the standard in that practice historically?
Oh boy, they tend to use flocculating chemicals to try to bind it up and precipitate it out of the environment, and those chemicals are just more toxic in the environment.
I thought it was enzyme based.
There are some some enzyme stuff that has been experimented with, but I don't know much about it.
Okay, thank you.
All right, I know. Okay.
He'll stick around.
Yeah, I'll stick around and I'll answer some more questions, but I really want everybody to meet and to coordinate. I know we're running over because everybody getting in, we started 15 minutes late, and I know that there's going to be a lot of questions, but I'm going to hand it over to V for a few minutes so I can rest my voice. And so V, it's like, how many years have you been in the Asheville area? 29 years. So a long-term resident of Asheville, did a lot of work in previous years with natural building, community organization, and so forth. So a lot of expertise in helping get communities organized and getting work done and so forth. And so what I'd like to do is just literally go through and meet the folks that are doing stuff on the ground. People have a lot of expertise really quickly. Let them all spend maybe about a minute or so introducing themselves and then go forward from there. So I'm gonna hand it off to V, and then I'll be around afterwards to answer more questions. So.
Who here has felt really overwhelmed by this whole situation? Raise your hand. And who here is feeling hopeful or some degree of hope? Yeah. So I want to just take a moment and have everyone look around. There's about 100 of us in the room, and this is essentially